Patterns of inheritance. Heredity is the transmission of traits from one generation to the next. Genetics is the study of heredity. Genetics began in the 1860s when a monk named Gregor Mendel deduced his fundamental principles by breeding garden peas. A heritable feature that varies among individuals, such as flower color, is called a character. Each variant of a character, such as purple or white flowers, is called a trait. Mendel studied what offspring would result if plants with purple flowers and plants with white flowers were cross-fertilized. The offspring of two different purebred varieties are called hybrids. The cross-fertilization itself is referred to as a genetic cross. A cross between a purebred pea plant with purple flowers and a purebred pea plant with white flowers is shown here. The purebred parents are called the P generation. Their hybrid offspring are the F1 generation. When plants self-fertilize or fertilize each other, their offspring are the F2 generation. Mendel saw that all of the F1 plants had purple flowers. By mating the F1 plants with one another, Mendel found that about 3/4 of the F2 had purple flowers and 1/4 had white flowers. In other words, there was a 3-to-1 ratio of purple to white. Alternative versions of genes are now called alleles. Alleles are alternative versions of genes that account for variations in inherited characteristics. For each character, an organism inherits 2 alleles of a gene, one from each parent. These alleles may be the same or different. An organism has 2 identical alleles for a gene and is said to be homozygous for that gene. An organism that has 2 different alleles for a gene is said to be heterozygous for that gene. If an organism has 2 different alleles for a gene, one allele determines the organism's appearance, and this is called the dominant allele. The other allele has no noticeable effect on the organism's appearance and is referred to as the recessive allele. Geneticists use uppercase italic letters to represent dominant alleles. Lowercase italic letters is used to represent recessive alleles. A sperm or egg only carries one allele for each inherited character because the 2 alleles for a character segregate from one another during the production of gametes in meiosis. This statement is called the law of segregation. When sperm and egg unite at fertilization, each contributes its alleles, restoring the paired condition in the offspring. Mendel's hypothesis predicted that alleles segregate during gamete formation. In the F1 plants, half the gametes will receive a purple flower allele, and the other half a white flower allele. During pollination among F1 plants, the gametes unite randomly. An egg with a purple flower allele has an equal chance of being fertilized by a sperm with a purple flower allele, or one with a white flower allele. Because the same is true for an egg with a white flower allele, there are a total of 4 equally likely combinations of sperm and egg. Punnett squares are used to highlight the 4 possibilities of combinations of gametes and the resulting 4 possible offspring in the F2 generation. 1/4 of the plants have 2 alleles specifying for purple flowers. 1/2 of the F2 offspring have inherited 1 allele for purple flowers and 1 allele for white flowers. These plants express purple flowers, which is the dominant trait. 1/4 of the plants have inherited 2 alleles specifying white flowers. These plants will express the recessive trait of white flowers. Mendel's model accounts for the 3-to-1 ratio that is observed in the generation F2. The ratio of plants with purple flowers to those with white flowers is 3-to-1. This is called the phenotypic ratio. The genotypic ratio would be 1 homozygous purple flowered plant, 2 heterozygous purple flowered plant, and 1 homozygous recessive white flowered plant. There is a relationship between alleles and homologous chromosomes. The matching colors of corresponding loci highlight the fact that homologous chromosomes carry alleles for the same genes at the same positions along their lengths. Each labeled band on the chromosome represents a gene locus, which is a specific location of a gene along the chromosome. Alleles of a gene reside at the same locus on homologous chromosomes. The two chromosomes may bear identical alleles or different alleles at any one locus. The organisms may be homozygous or heterozygous for the gene at that locus. A dihybrid cross is a cross between two organisms that are heterozygous for two characters. Mendel crossed homozygous plants having round yellow seeds with plants having wrinkled green seeds. The union of these gametes from the P generation yields hybrids that are heterozygous for both characteristics. These are known as dihybrids. These 9 genotypes will produce 4 different phenotypes in a ratio of 9:3:3:1. There are 12 plants in the generation with round seeds, compared to 4 wrinkled seeds, and 12 yellow-seeded plants, compared to 4 green-seeded ones. Notice that 12 divided by 4 reduces to 3:1. A dihybrid cross is thus equivalent to 2 monohybrid crosses occurring simultaneously. Let's look at the independent assortment of genes in Labrador retrievers. Suppose you have a chocolate lab. You can tell its genotype must be homozygous recessive, since that is the only combination of alleles that produces a chocolate-coated phenotype. But what if you have a black lab? A black lab could have 2 possible genotypes, heterozygous or homozygous. There is no way to tell which genotype is the correct one by just simply looking at the dog. To determine your dog's genotype, you could perform a test cross, which is a mating between an individual of a dominant phenotype but an unknown genotype, and a homozygous recessive individual. In this case, a homozygous recessive chocolate lab. If the black lab's parent's genotype is homozygous dominant, we would expect all of the offspring to be black, because a cross between genotypes homozygous dominant and homozygous recessive can only produce heterozygous offspring, showing the dominant trait. Geneticists who study people obviously cannot control the mating of their research participants. Instead, they must analyze the result of matings that have already occurred. First, a geneticist collects as much information as possible about a family's history for a trait. Then the researcher assembles this information into a family tree called a pedigree. Here we see a pedigree tracing the incidence of freckles versus no freckles, a trait that is believed to be controlled by a single gene. The capital letter F stands for the dominant allele for freckles. The lowercase f symbolizes the recessive allele for no freckles. The dominant phenotype, freckled, results from having either 1 or 2 dominant alleles. If both alleles are recessive, the phenotype is then recessive, having no freckles in this case. In pedigrees, unshaded square symbols represent male. Unshaded round symbols represents a female. Colored symbols indicate that a person does not have the trait being investigated. Unshaded symbols represent a person who does have the trait. The earliest or oldest generation is located at the top of the pedigree, and the most recent at the bottom. By applying Mendel's laws, we can deduce that the allele for no freckles is recessive, because this is the only way that Karen does not have freckles, even though both of her parents have freckles. Dominant alleles are not always more common in a given population. For example, the absence of freckles is more common than their presence, even though the presence is dominant. Dominance means that the heterozygous genotype results in the dominant phenotype. Traits that are more common in nature are called wild type. Wild type actually has nothing to do with dominance. A recessive trait may in fact be a more common trait found in a population. Most genetic mutations that cause medical disorders are recessive. Most people who have recessive disorders are born to normal parents who are both heterozygous. That is, the parents are carriers of the recessive allele for the disorder, but appear normal themselves. Suppose two heterozygous carriers of albinism have a child. 1/4 of the children of this couple are likely to display albinism. 1/2 will be carriers, and 1/4 will have normal pigment and will not be carriers. A disorder caused by a dominant allele is called achondroplasia. Achondroplasia is a form of dwarfism in which the head and torso develop normally, but the arms and legs are short. The homozygous dominant genotype causes the death of the embryo. Heterozygous individuals have only a single copy of the defective allele and have the condition achondroplasia. A person with achondroplasia has a 50% chance of passing the condition on to any children. Therefore, all those who do not have achondroplasia, which is more than 99.99% of the population, are homozygous for the recessive allele. This example makes it clear that a dominant allele is not necessarily more common in a population. For some characters, the appearance of the hybrids falls between the phenotypes of the two parents. This effect is called an incomplete dominance. For example, when red snapdragons are crossed with white snapdragons, all the F1 hybrids will have pink flowers. In the F2 generation, the genotypic ratio and the phenotypic ratio are the same, 1:2:1. Hypercholesterolemia is a condition that causes dangerously high levels of cholesterol in blood. Unaffected individuals are homozygous dominant. Heterozygotes have blood cholesterol that are within about twice the normal amount. The dominant allele specifies a cell surface receptor protein that liver cells used to mop up excess low-density lipoprotein, or LDL, known as bad cholesterol, from the blood. With only half as many receptors as homozygous dominant individuals, the heterozygotes can remove much less excess cholesterol. The ABO blood groups in humans involve 3 alleles of a single gene. Various combinations of these 3 alleles produce 4 phenotypes. A person's blood type may be A, B, AB, or O. A person's red blood cells may be coated with carbohydrate A, which gives them blood type A. If they're coated with carbohydrate B, their blood type is B. If the person's red blood cells are coated with both carbohydrate A and B, their blood type is AB. And if a person's red blood cells does not have either carbohydrate A nor carbohydrate B, they are type O. There is a clumping reaction that occurs between the antibodies and foreign blood cells based on blood typing. This shows that there is an adverse reaction that can occur when someone receives a blood transfusion of incompatible blood. Matching compatible blood groups is critical for safe blood transfusions. If a donor's blood cells have a carbohydrate A or B that is foreign to the recipient, then the recipient's immune system produces blood proteins called antibodies that will bind to these foreign carbohydrates and cause the donor blood cells to clump together, potentially killing the recipient. A and B are co-dominant with one another. A person inheriting both A and B will have a blood type of AB. Both A and B are dominant to type O. A person with AO blood will have type A. A person with BO blood will have a type B blood. O is recessive, and only a person receiving 2 recessive alleles will have the blood type O. Type O blood reacts with no others, making it a universal donor. Conversely, a person with type AB is a universal receiver. In many cases, one gene influences several characters, a property called pleiotropy. An example of pleiotropy in humans is sickle cell disease, sometimes referred to as sickle cell anemia. In sickle cell disease, the sickle cell allele causes red blood cells to produce abnormal hemoglobin proteins. These abnormal hemoglobin proteins tend to crystallize and change the normally disc-shaped red blood cells to deform into a sickle shape with jagged edges. These sickled cells can lead to a cascade of symptoms such as weakness, pain, organ damage, and paralysis. Because of these cell shapes, these sickled cells do not flow smoothly in the blood and tend to accumulate and clog tiny blood vessels. Blood transfusions and drugs may relieve some of the symptoms, but there is no cure for sickle cell anemia. Sickle cell disease kills about 100,000 people in the world annually. Many features such as skin color and height result from a polygenic inheritance. Polygenic inheritance refers to the additive effects of 2 or more genes on a single phenotypic character. For example, researchers have identified over 180 genes that affect human height. A gene located on sex chromosomes is called a sex-linked gene. Red-green color blindness is a common sex-linked disorder that is caused by a malfunction of light-sensitive cells in the eyes. A colorblind male has offspring with a homozygous female with normal color vision. All of the children have normal color vision. All of the females will be carriers. A female carrier mates with a male who has normal color vision. The classic 3:1 phenotypic ratio of normal color vision to colorblindness appears among the children. All the females have normal vision. Half of the males are colorblind. Half are normal. Because the color blindness allele is recessive, a female will only be colorblind if she receives that allele on both of her X chromosomes. For a male, however, a single copy of the recessive allele confers color blindness. For this reason, recessive sex-linked traits are expressed much more frequently in men than in women. Thank you for watching.